Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Shervin Takyar. He's an associate professor at the Yale School of Medicine. He has a PhD in microbiology and molecular biology from University of Queensland, Australia. And he's worked on several lentiviral vectors, RNA, etc. Today we're going to talk about microRNA in the lungs and what is its role? How does it impact inflammation, lung injury, and cancer? So, Shervin, thanks for coming.
Thank you for inviting me.
Yeah, so how would anyone even know that there's microRNA in the lungs? How is this first found and why?
Yeah, so microRNAs actually came about from a study on worms. There was a number of there were a number of scientists. They had this astute observation that parts of the genome that are not really classic genes, they do not really code a protein, seem to have a significant effect, a significant cellular, molecular, or organic effect in these organisms. And it was a very difficult thing to have people understand it or accept the concept. But it took around 10 years, really, for people to understand it and accept it as a concept in biology. But then after that, suddenly everybody rushed to figure out what they are doing, because it was shown that these microRNAs are a different type of language. They are part of a language that cells use to talk to each other, and no eukaryotic cell that people looked at didn't have them. They were all over. So, the fact that lung cells also have them, is not a big surprise. Every cell has it, and they use microRNA as a tool to talk to each other. And that's the most genes talk to each other through microRNAs, cells talk to each other through microRNAs. It's part of the language. If you're decoding or computer work, software work, there are different levels of language. microRNA is probably one level above genes. And
one question: when you say that cells use them all the time, are they expressed in extracellular vesicles, or how are they used? Where do they appear?
They can also be packaged into extracellular vesicles. That's one of the ways that cells can send them to each other. But the more complete work is inside each cell. So, for instance, if a cell wants to communicate with another cell, a lot of times it's not just one factor, it's not just one message, it's a number of messages. And we have seen this in nature again and again, right? One action takes more than one gene. So, for instance, if one cell is telling another one, get activated, that is not just sending or releasing one cytokine. It's a group of cytokines that are released at the same time. Now, microRNAs, we know for sure, inside the cell, control a group of genes that then are being expressed and released as that message. So that's how I'm saying there are higher levels. Also, it has been shown that these microRNAs are packaged into vesicles and sent to another cell. So there are multiple levels. They're hard to decipher completely. The magic of microRNA is that they can control many genes or different genes at different times and different genes at different times. So it's a language by itself.
So what happens when microRNA gets into a cell? What does it typically do? Where does it go? And what do they translate into?
Yeah. So what we know as of now is that if microRNAs get into a different cell, like for instance, if they're packaged and go to a different cell, they can use the same machinery that they use in the cell of origin. It seems like the machinery, the response machinery that is in R&D cells are very similar. This basically the same. They call it RNA-induced silencing complex. And to tell you the truth, I mean, RNA-induced silencing complex, which is a conglomerate of molecules, this also works in plants. And for another group of small RNAs that we call siRNAs or small interfering RNAs. So the machinery is that machinery takes one microRNA and then matches genes with it. And the genes that are matched with a microRNA are inhibited. So suddenly this machine in the cell of origin or in the target cell gets a microRNA in, matches genes with it, and inhibit those genes. And seems like the type of gene, the number of them depends on the context, right? You don't want the machine working all the time or some genes being on and off all the time, right? Things change.
Do microRNAs only inhibit, or do they upregulate as well?
So most of the information, maybe more than 95% of information that we have about microRNAs is inhibitory, is that they have a role in inhibition of genes. They do also. There have been instances that show that microRNAs may increase the expression of genes, and that seems to be dependent on the components of that risk machine that I talked to you about. But as I said, the majority of literature and findings is about inhibition. So I don't know if you're familiar with promoters. So promoters are factors that increase the expression of genes, transcription genes, a number of them. So microRNAs are the opposite end. It seems like these two work together and fine-tune the expression.
Do they preferentially inhibit certain functions and not others?
Yes, exactly. That is the power of microRNAs. Since it is they are matched based on the sequence that is in microRNA, they bind to certain genes that have the receptor, if you will, or the complementary sequence. They only bind to those genes and not to others, and that's where it comes from, right? They can select, and the good part about it is it's all sequence dependent. So if you study the sequence of microRNAs, you can predict to some degree which genes are going to be inhibited. The interesting part is that it is not a full complementarity, right? MicroRNAs have this degenerate, quote unquote, nature, meaning that they are not a complete complementary sequence, and that gives them the possibility of controlling a number of genes with close sequence similarity. And that's where the selection comes from.
So is there a family of genes that, again, the microRNAs tend to target ones that are involved in, this or that or the other?
Yes, depending on the context. So if the question is, are there any particular group of genes and other group of genes are not affected? No, that's not true. Any genes can be affected based on its sequence and based on its expression pattern. In a way, microRNAs are telling us, forget about your classification. the sequence of these microRNAs and the context of the cell and what is being expressed, being transcribed, will dictate what is inhibited when. Basically telling us, okay, you want to classify like we do. We classify.
So what are some of the what are some examples? What gets inhibited and what tissue?
All right. Now, for instance, so I can tell you a little bit about our work, the work that we are doing or we did and published. This was on the inhibition of a number of endothelial genes. I mostly work in endothelium. So the work started from an observation that was made years ago and was an odd observation that a vascular growth factor was high and in patients with asthma, right? So when you talk about asthma, most of the time you talk about, lung epithelium or immune cells. That's the focus of research mostly. But for years, people have been seeing that this vascular growth factor, VEGF is called, is up in people who have severe asthma. And then my former PI and Dr. Elias and Chun-Gun Lee, who was working in his lab at the time, they defined this in an animal model, animals and cells, that this vascular growth factor by itself can induce an asthma phenotype. So that's where I started. And my question was, okay, well, can that be translated to a microRNA? Like, does this vascular growth factor affect a microRNA in endothelial cells? Or it's just a growth factor that affects, immune cells? And that has been the last 12 years. So I started from cells, went to animals, then went to humans, and showed that there is a correlation between this VEGF and a particular microRNA, microRNA1. Now, I wanted to see if some of the downstream effects of VEGF can be explained by this microRNA. People knew that, for instance, when VEGF goes up or VEGF is added to a cell in culture, some genes are expressed higher and some genes are expressed lower. This microRNA, microRNA1, we found over the last 10, 12 years that makes a link between these two observations. VEGF goes up, and for instance, one of the genes we know about, P-selectin, that is very important in adhesion of eosinophils, goes up. The link between VEGF going up or being added and P-selectin going up is a microRNA in between. That microRNA is microRNA1 that comes down. So VEGF goes up, microRNA1 comes down, P-selectin is released from inhibition, if you will, and is expressed.
So that makes sense. If you're saying microRNAs are inhibitory,
exactly.
microRNA going down would mean less inhibition.
Exactly. In this work, we found 3 more genes apart from P-selectin that are inhibited by microRNA1. And 3 of them were known before that have some correlation with VEGF, and one of them was new, which, some effects, some cellular effects of VEGF can be explained by this new gene that nobody thought belongs to this family and is targeted by this microRNA. The gene is MPL that we published. And that's why I'm saying, microRNAs are telling us, forget about your classification. It's the function and the context and how we work.
How would you use microRNA changes and what we know about particular genes and other factors to triangulate? I guess that's what you're saying. We could probably use this to triangulate effects that we wouldn't normally see. It's just another variable, another factor we can look at, right?
Exactly. So for instance, there have been a number of things like this, right? When we have come to an age where we know targeting a single gene a lot of times does not work. We know that. And the reason why it doesn't is that when a cell or an organ is in a specific reaction or a specific state, it expresses a number of genes. So if you target one of them, like VEGF we were talking about, there are anti-VEGF drugs available and in use in cancer, for instance, because angiogenesis in cancer or vasculature in cancer is very important.
Right.
Blocking VEGF in cancer a lot of times does not give the effect that you want, and the cancer still grows because tumors are very smart or resilient. They come up with other genes. They come up with other genes to do the same thing through another pathway, right? So blocking one gene a lot of times does not work. And I tell you, the majority of the molecular biological work right now is based on single genes. We are just targeting.
So are you thinking that microRNA may be more of a lever or more of a factor or stronger than trying to knock out a gene. Or
yes, exactly. I think they're one step closer to inhibiting a group of genes, to modifying a group of genes rather than one. So if cells, if you inhibit one gene, they come up with another gene that does the same thing in the same pathway. Now targeting a microRNA will give you this possibility of targeting a group of them, so that maybe you're closer to inhibiting a response rather than a single gene.
Well, there's a lot of redundancy in the body, which, is for resilience, I would guess.
Yes.
So it makes sense that, you can't just always knock out one gene or just use one molecule even to accomplish a task.
Exactly. And there have been some, semi-philosophical articles about microRNAs based on observations, how they actually, it's very interesting. They use, the authors have used the same word, redundancy, how microRNAs are used in redundant circuits. They bring about this redundancy, or how it is done, since they are affecting a number of genes. When you target one, another one works through a loop, through a microRNA.
Mm-hmm.
Yeah.
Well, so this makes sense. I mean, again, I've heard microRNAs are predominant in extracellular vesicles. So
yeah, so it has been. So I tell you, microRNA work has two. I mean, let's talk about the social aspect of it. Maybe, I don't know, the social scientific part of it. So microRNAs are small and they are resilient in a sense when we talk about degradation. Among RNA scientists, we know that, for instance, a group of people cannot work with microRNA. A certain type of phenotype cannot work with RNAs, not with microRNA, with RNAs. There's a lot of degradation going on in the labs, and that's one of the difficulties of RNA work. MicroRNAs are actually pretty small, and they do not degrade as much as RNAs, as messenger RNAs. So they can be measured easier. So the point of all this was they actually can be measured easier. Right? They do not.
What's micro about them? Is that they're just not very long series of sequences?
Their size, yes. Like messenger RNA can be anywhere between 600-700 base pair to up to like 8-9 kilobase. MicroRNA is only 22 nucleotides.
Wow, really?
So they are very small. They are very small. Because they're small, they can be measured, and techniques were developed 10 years ago that can measure them accurately. Actually, there are not many, but there are a couple of techniques that are very good at measuring microRNAs. So people can measure them easier, right? So then we have this whole microvesicles or exosome work that came about maybe around like 5-6 years ago, 5-6 years after the microRNA work. And in these vesicles, one of the first things that people could measure was microRNA. And it made sense, right? If you are sending, based on all the conversation we had, if you're sending a message to another cell, you can control a number of genes in another cell by a microRNA, rather than just sending one cytokine. So that's how suddenly the role of microRNAs in microvesicles got so much press, because they could be measured. And that's what we always face in science. What we can measure, we talk about a lot. Doesn't mean that nature also gives the same importance to it. But it has been shown by a few good works shown that microRNA goes to a target cell and affects the translation or gene expression in those cells also.
So, at the beginning of the conversation, the topic was microRNA in the lungs. So where are you finding it? Why is it important? What role does it appear to have?
Yeah, so as I told you, since the previous work in our group was on the vascular growth factor, my question was, and I was a basic scientist at the time. I was a clinical fellow, but I was a basic scientist in my other life. So I just went very simple, and I said, I asked the simple but unpopular question that, well, what's the role of vasculature then? If you're talking about asthma, right? I'm a pulmonologist in a pulmonary section, talking all the time about endothelium, about vascular cells. And I wanted to show, and then I went to this literature about microRNA and how they work. So I think, and then I showed through models, through different methods, that changing a microRNA, specifically isolated change of a microRNA in endothelium, can affect asthma or allergic inflammation. So, as I said, majority of work in asthma is not on vasculature. It's on immune cells and then lung epithelial cells, some of that. What I'm bringing to the table, together with some researchers, is that endothelial cells matter in asthma. If you and I were talking about asthma literature, we would probably, out of a 1 hour conversation, we will have 2 sentences about vasculature. It's a part that has not been talked about a lot. But my rationale was that, we're talking about immune cells, right? These immune cells, where do they come from? They come from the blood. They have to get into the tissue through the blood. And the gatekeeper is an endothelial cell. So there has to be a very strong gene expression pattern in the endothelial cell. So the first focus of this work is an endothelial gene is having a big part in allergic inflammation. So I would say that is the first message. And that's how we looked at this, the
site of, I guess I'll call it the site of insult. You breathe in something and your lungs don't like it. So I'm calling it the site of insult. Does that happen at the endothelium initially?
So that is, of course. So some of it does. We think that we still think that the majority of that insult in something like asthma, or let's say that trigger, let's call it trigger for now, that the majority of that is in epithelium, because that's the first line of defense. That's the first, wall against an antigen. But then epithelium, we think, talks to endothelium. So that's one paradigm, that the trigger affects the epithelium or immune cells that are like macrophages that are in the area and take the message, and then they can talk to endothelium or get into the vessels and go. Now, I should modify the sentence by saying that what you said about damage, right? Damage can happen to endothelium. Trigger can happen to endothelium also. And that is ongoing work. There are factors that are affected in the endothelium by exposure to a challenge. And it's a little hard to say, is it all direct or is it through epithelium? We know that endothelium is affected by a large degree. The current crisis with coronavirus, for instance, or the situation that we have, is mostly an endothelial phenomenon in the lung. The factors, the response is an endothelial phenomenon. Epithelium gets damaged, but endothelium causes it. So there is evidence that endothelium gets affected, gets triggered, but in allergic inflammation, it has been mostly behind the scenes and not paid attention to as much as epithelium and immune cell.
Do you think that microRNAs are a huge part of the signaling that goes on? Let's say, again, there's the trigger happens in the endothelial cells, and then they're sending out, EVs with microRNA in them to signal the immune system, or microRNA is just playing that role in general.
Well, I am just going based on the observation in this, and that was my question around 9 years ago when I started this particular work. But I changed this microRNA, this one microRNA, only in the endothelium of an animal model. That's all I did. I just changed one microRNA in the endothelium of an animal model. And I found that the allergic response, the asthma type response, comes significantly down, is significantly decreased. So that's the observation. Now, I'm also showing it is affecting, for instance, eosinophil transit into the lungs, right? But those are secondary to the single change of microRNA in endothelium. So, that's the observation.
How are microRNAs obtained? How do you know where they are and how to harvest them and that they're the right ones?
Yes, so that was part of it. The techniques we worked on and developed. What I started was isolating the endothelial cells based on their surface markers. And when I started, I could isolate three types of cells, three types of immune cells, epithelial cells, and endothelial cells. All those are those experiments were done based on immune sorting, meaning with antibodies, we sort the cells based on their surface markers. And then in those cells, I measured specifically the mature, we call them mature microRNAs. Now, as a side, microRNAs go through different stages of maturation in the cell, and they are very interesting the way they are affected and matured. So I measured specifically the mature microRNA, which is the effective type, the one that the effector type. I used these two techniques. One of them was sorting of cells, and second was a specific measurement of the mature microRNAs. And then based on that, I measured the response, the specific response in endothelium in different states, with VEGF, without VEGF, in asthma, different kind of. But yes, so that we developed very early in the work. I developed that in the lab, and we went on from there based on these two techniques.
Where do microRNAs? What's the biogenesis of them? Like, where do they come from, and are they packaged in any way normally inside the cell?
Yes. So microRNAs, so far what we know is that they are very similar to classic genes. They are transcribed from pieces of DNA, from parts of DNA. This is again they probably classified as the same, junk DNA, quote unquote, that people talked about for ages, that are not classic genes. They cannot be defined as classic genes, but they're stretches of DNA. They are transcribed from DNA into RNA, and then a number of enzymes inside the nucleus and then outside the nucleus in the cytoplasm work on these different precursors, right? It's the long precursor, a prime microRNA, and then that gets worked on by an enzyme and then is transferred into the cytoplasm. Another enzyme works on it, and then it matures into the small RNA that we call microRNA. Happens in the nucleus and in cytoplasm in different machines, inside different machinery, which is by itself a very interesting a pathway and worked out. And some of them have even crystal structures of how they work and how they can measure and cut and prune and all of those things for a specific message. They're all parts of the machinery of the cell.
So what's how big is your goal? Do you just want to understand them in the context of the lungs with asthma, or do you want to go more broad and, in general, characterize microRNAs and their roles and all kinds of functions?
Yeah, no, I already, in 2017, I published this work and a part of this pathway in cancer. So I'm more of a basic molecular biologist. And to me, I think this is a way, this whole paradigm is showing us a way to understand connections that we were not understanding before. Which gene, which context, and which response. And I think that's the interesting part from a basic science. I'm also a clinician, so I do see asthma patients. I have a lot of COPD and emphysema patients. I take part in the care of cancer patients. So yes, so I am trying to find if if microRNAs, or what part they play. I think the fact that they play very important roles is out of question. We all know that they play very important roles, but which specific genes in which context is very interesting, and I think very important from a medical point of view. So one part of the work is that I published in Cancer. This is in asthma. We also working on lung injury, which is nowadays is pretty famous. This ARDS after a viral infection is a type of lung injury in endothelium. I am also have been working on the question that you asked. How are they matured and pruned and made and expressed? So I'm also looking on the machinery that specifically brings them about. Parts of this pathway, as I said, is known. Enzymes like dicers are known to affect microRNAs, but we are finding just recently that for specific types of microRNA is beyond that. It's not just dicer. There are molecules downstream, machinery downstream of dicer that affects the levels of microRNAs. And we are doing experiments on those to define the exact pathway. For instance, I just told you about dicer, very important enzyme in maturation of microRNA. You can inhibit dicer, and very interesting things happen to the cell, but still a number of microRNAs work. So that doesn't make sense. There has to be other enzymes involved. And lo and behold, we have found other enzymes that are working on this pathway. So that's one direction that I'm going. The other direction is different diseases, as I said. And then, how other RNAs talk to microRNAs. I told you microRNAs is one step. They are one level above the genes. It's interesting that since like 8-9 years ago that we know there is a higher level. It's called competing endogenous RNA or CERNA. This is different RNAs in the cell talking to each other, not just microRNAs in genes. There are other non-coding RNA that are bigger. They're not small, but they don't code any genes. They seem to be talking to messenger RNAs, to microRNAs. So a lot of conversation going on in the cell. We are trying to listen in through the RNA,
Yeah, it's amazing.
Listen in.
These RNAs are just a strand of base pairs. I mean, what other features do they have on them that are unique?
What you just said. They're a strand of base pairs. So RNA exists as a single strand. Parts of the RNA base pairs on itself. So the majority of RNAs are like that. There are double-stranded RNAs in the cells also, but what we know about that and what they do is much more limited. A lot of RNAs in the cell are single-stranded. Parts of the molecule is double-stranded because it base pairs with different parts of itself and has a structural role. The work that I did before with Harry Noller was, for instance, he was one of the people who brought about this whole RNA world and how RNA works, and he was one of the group that showed RNAs are molecules that can be both structure and function, can act as enzymes, can act as structure by base pairing and making different types of modules as a structure. For instance, the ribosome has a lot of RNA structure in it. Also can work as enzyme, work on ribozymes, if you've heard. They are RNA enzymes, and here are microRNAs that are not even coding any genes but affecting other RNAs. So yeah, they're fascinating. There are these molecules that have many different function, many different facets, if you will, and I think there is a lot really to be learned from the way they behave.
Oh, okay. Well, what's the best way, Shervin, for people to find out more about your work and to follow you?
So I published this paper in a Journal of Allergy, Clinical Immunology. I should tell you, I do not spend a lot of time making websites and this and that, but I probably will try. to put some of it out, just because a lot of people, patients, colleagues say that we have to work on this. So I have a website at Yale that I have put some of this information in, and then through media like you, like this conversation. I think I'm willing to try to simplify and have the information out. And quite honestly, since I'm very focused and I do clinical work and research, there's not a lot of time remaining. So I try my best, but I've been told by a group of people that I should work a little bit more on putting the information out in simple terms. And that's why when I got your invitation, I thought it's a good platform to go out and have a conversation.
Yeah, well, the best communicators, they do it simply. And I could tell you, for instance, I've seen, it took me, a year of wanting to read and understand scientific papers to be able to start reading them. It took a lot of effort, and I always appreciate when a scientist comes out with a book that is consumable by the public. I see a lot of them come out with books and they list it as a textbook. Who's going to read that just with that label? Yes. And it's $150 or something.
Yes.
So there's a lot of basic things I think scientists can do to make it accessible to the public. And you're not necessarily dumbing it down, and you're calling more attention to it. And why not?
Yes, I think you're right. I think you're absolutely right. I think it's a time that we have to. I tell you, I have probably 4 or 5 books next to the bed to read, and there are scientific books. Scientists, like very serious scientists, have written them, and they are accessible. And I mean, I read lay science in other fields, and I really like it. And I think you're absolutely right. I think we are not doing enough of this. We are not, like microRNA work has been around for now 20 years, right? We have not talked enough about that. And I like to be part of the effort. I'm seeing how important it is to have people know. I don't know if you know this or not. When we write grants, there is a part where we are asked to write the proposal in maybe 5 sentences for lay people, for non-scientists. And I tell you, that part is one of the hardest. But I think it is. I think to write those 5 sentences in a way it makes sense. But I think it is energy well spent. When I go through that exercise, I come to the conclusion that no matter what you do, saying it in a way that everybody can understand has a high value. You probably are familiar with Kurt Vonnegut, right?
I've heard of him.
Yes.
I haven't really read his stuff, but I've heard of him.
Yeah. So in one of his books, he's talking about the scientist, and the scientist says that any scientist who cannot explain his work to a 5-year-old in like 3 sentences is a hoax. I read this a while ago, and I thought it's what an interesting concept that, if you are really into your science and you know what you're doing, you should be able to explain it in simple terms. But it's not easy. And I like to try. I mean, you're just saying this, bringing it up. I think maybe I'll start writing something simpler.
It's okay. I mean, you could do it in layers and stuff, but it's all right. Well, that's part of the translation here is me asking questions and you're explaining. So you did a good job.
Sure.
Well, very good, Shervin. Thank you for coming, and you gave that resource, and I appreciate you being here.
Thank you, and thank you for inviting me and being a media for science to be talking to people directly.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.